Ja, det var bra. Man vill gå förbi och sätta sig. Vad vill du? Vi ska göra. Ta en av stolarna där framme och ställa här. Bra. Jag kan ta den också. Ja, den där är jättebra. Ja, okej. Hej Lotta. Det var väldigt bra. Vad vi skrev på svenska. Där är advanced information. Jag tycker det är bra att stoppa det här. Ni kan ju bara dela upp det där. Ska någon med Maria vara med och dela ut också här? Ja. Det går inte. Då kommer det stå nej, vi är på bilder då. Ja, här har vi ju lättare att fiska. Här har vi lättare att fiska. Nu ska vi ganska nog. Ja, det kan har vi alla platser.
What will happen now? This looks good. So, ladies and gentlemen, good morning and very welcome to Nobel Forum for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman and I'm the Secretary General of the Nobel Assembly. I will first read the announcement in Swedish, followed by English. And then we will go on to present background to the prize and open up for questions. Så då kör jag igång. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2024 skall delas lika mellan Victor Ambros och Gary Ruvkun för upptäckten av microRNA och dess roll i posttranskriptionell genreglering. Och på engelska. The Nobel Assembly at Karolinska Institutet has today decided to award the 2024 Nobel Prize in Physiology or Medicine jointly to Victor Ambros and Gary Ruvkun for the discovery of microRNA and its role in post-transcriptional gene regulation. Here are the two laureates a few years ago. Victor Ambros was born in Hanover, New Hampshire, in the US in 1953. The work leading to today's award was performed at Harvard University, and he's currently a Silverman Professor of Natural Science at the University of Massachusetts Medical School. Gary Ruvkun was born in Berkeley, California, in the US in 1952. The work leading to his Nobel Prize was performed at Massachusetts General Hospital and Harvard Medical School, where he's now a professor of genetics. I will now turn to the Nobel Committee Vice Chair, Olle Kempe, who will describe the discovery. So please, Olle.
Ladies and gentlemen, this year's Nobel laureates have discovered microRNA, a tiny molecule that has opened a new field in gene regulation. Genetic information is stored in DNA in the cell nucleus. This genetic information is copied into mRNA, and mRNA is then translated to protein. Proteins are the building blocks that are responsible for major functions in our body. For example, neural function, muscle contraction, absorption of nutrients in the gut, and defending us from invading pathogens by immune cells. Different tissues express different proteins with different functions. Since the DNA in our cell nuclei contains exactly the same coding information in all cells, the question is, what determines that only the right genes are transcribed into mRNA and then translated into the correct tissue-specific proteins at the right time? A first important step in gene regulation is governed by DNA-binding proteins, so-called transcription factors, ensuring that only the correct genes are transcribed into mRNA. As a result, the correct proteins are made in each specific cell type. In fact, for many years it was believed that the main principles of gene regulation had been elucidated. The mechanism would, however, turn out to be more complex. For this year's Nobel Prize, an elegant little organism was the key. The tiny C. elegans has been used as a model for more than half a century. The worm contains only 1,000 cells, but still forms many of the different tissues as humans do, including a nervous system, a gut, and muscles. Researchers had previously found two interesting C. elegans mutants with timing defects during development. The Lin4 mutant, with a red label as shown on the slide, becomes larger because one developmental program from egg to an adult worm is erroneously repeated. In contrast, the Lin14 mutant, as shown with the blue label, skips some stages during development and becomes smaller. The Lin4 and Lin14 mutants have strikingly opposite appearances, and although the genes had not been cloned at the time, Victor Ambros was able to show that Lin4 negatively regulated Lin14. This year's Nobel laureates, Victor Ambros and Gary Ruvkun, were intrigued by these phenomena and decided to attack the problem, identify the genes, and understand how these genes work. Victor Ambros started an effort to clone Lin4. Over several years, he made painstaking efforts to home in on the exact position of the gene. To the amazement of Victor Ambros, when he finally cloned the Lin4 gene, it only produced a tiny RNA that could not be translated into a protein. Concurrently, Gary Ruvkun succeeded in cloning the Lin14 gene, which does translate into a protein, as shown in the box on the left. During early development of the worm C. elegans, the Lin14 protein is made, but the production of Lin14 is suppressed by Lin4 during the later stages of development. Gary Ruvkun was intrigued when he found that the negative control of Lin14 by Lin4 seemed to involve the terminal segment of the Lin14 mRNA that was not translated. As depicted on the box on the right side, he observed that deletions of this region abolished the effect of Lin4, and therefore he postulated that the regulation might occur at the mRNA level. The observations by Ambros and Ruvkun were equally puzzling. How could these two so very different molecules interact with each other? As Victor Ambros and Gary Ruvkun knew each other from their days as postdoctoral fellows, they decided to exchange information about the Lin4 and Lin14 genes. On comparing their findings, it turned out that part of the sequence of the small Lin4 RNA matched complementary sequences of Lin14 mRNA in the terminal region that does not code for a protein. They then understood that the binding of Lin4 to Lin14 mRNA prevented the production of the Lin14 protein at the level of mRNA. At this point, they had discovered a novel and unexpected mechanism of gene regulation, microRNA. For a long time, however, microRNA was believed to be an oddity peculiar to C. elegans. This perception changed 7 years later when Gary Ruvkun, in the year 2000, identified a second microRNA produced by the LET7 gene in the world. Unlike GLIN4, LET7 was found to be highly conserved and is present in human and most of the animal kingdom. This finding made the previously dormant microRNA explode. As it turned out, many microRNAs are highly conserved throughout evolution over several 100 million years. In more complex organisms, a larger number of microRNAs have evolved, with humans having more than 1,000 distinct microRNA genes. Today, we understand that the majority of all genes are regulated by microRNAs. Every microRNA regulates several genes, several mRNAs, and each mRNA is often regulated by many distinct microRNAs, creating a robust system for gene regulation. The seminal discovery of microRNA has introduced a new and unexpected mechanism of gene regulation. MicroRNAs are important for our understanding of embryological development, normal cell physiology, and diseases such as cancer. As an example, tumors often perturb microRNA networks in order to grow. Victor Ambros and Gary Ruvkun have jointly been awarded this year's Nobel Prize in Physiology and Medicine for their discovery of microRNA and its role in post-transcriptional regulation. I now hand back to Professor Thomas Perlman, the Secretary General of the Nobel Assembly.
Thank you very much, Olle. So with this, we would be happy to answer questions. And you've already been introduced to Professor Kempe, but on my side, I also have the chair of the Nobel Committee, Professor Gunilla Carlsson-Hedestam, and member of the Nobel Committee, Professor Rickard Sandberg. We have a question up there first.
Okay, thank you. I'm from TV4. Do I need a microphone? No, maybe not.
Yes, please.
I have a question regarding your second last slide, if you could get back to that one. I wonder how it's possible for the microRNA to get to the mRNA first, so to speak, so that it shuts down this mRNA and the protein is not made. I mean, if the mRNA is made first, then the protein would be made anyhow. And I also wonder what the different cell types, why is the microRNA, a special microRNA, active in one sort of a cell and not in another sort of a cell?
Yes, these are quite advanced scientific questions, and I'll turn to Professor Rickard Sandberg.
So regarding the first question, so we skipped a lot of detail in this presentation, but the microRNA strand is part of a protein complex that finds the mRNA in the cytoplasm. And there it can recruit other proteins that help shorten the poly-A tail, which leads to either translational inhibition or mRNA degradation. The second question about microRNA genes, I mean, they are also transcribed, so they're copied from the DNA into their RNA form, They are processed, and that can be done cell type specifically. So a specific microRNA will end up in a muscle cell, whereas other microRNAs end up in, for example, nerve cells. And because of that, they can have cell type specific effects on the mRNA.
Hey, my name is Stefan Trump from the German news agency dpa. Thomas, it's early morning in the US, so my question would be, have you already reached one of the laureates? And yeah, what did they say for being awarded?
Yeah, nowadays it tends to be a little more difficult. People have mobile phones on silent, but I was able to wake up Gary Ravkan. So I think he had a landline. I was able to reach him. His wife answered, and it took a long time before he came to the phone and sounded very tired. But he quite rapidly was quite excited and happy when he understood what it was all about. He was so enthusiastic, and his wife too, so he handed over the phone to his wife, who also wanted to talk to me for quite a while, and they were thrilled about the prize and coming to Stockholm in December. I was not able to reach Victor Ambros yet. I left a message on his mobile phone and hope he gives me a call soon.
Hello, my name is Bogun Radomski. I'm from the Polish television. A question I have is